Cell raft technology has the capacity to accelerate the process of monoclonal cell line development when you compare it to some of these technologies that depend on dispensing a single cell alone into a well. Our innovative culture dish, the cell raft array, enables flask-like culture with single-cell spatial separation. So your cells will grow on this culture dish much in the same way and at the same rate that they would grow in bulk culture, yet they're maintaining single-cell spatial separation. So since they're continuing to grow and sharing that same reservoir of media, they're continuing to communicate with each other, secreting growth factors and cytokines that help those cells maintain really good viability and growth on the array. So this speeds up the process of going from a single cell into a clonal colony of cells. Then with our system, you're able to capture and retrieve those clonal colonies of cells directly off of the cell raft array and culture those in 96 well plates. And the outgrowth is much more efficient because you're starting with a clonal population of cells that you've documented as monoclonal from our cell raft array into a 96 well culture dish. Since the entire array can be imaged effectively and rapidly using our cell raft air system, this means that the bioprocess scientist is able to maintain a record and easily document the process of the cells as they grow from one cell into two cells and then into a clonal colony prior to selection of that clonal colony directly off of the cell raft array for downstream propagation. By isolating clonal colonies of cells that have overcome that barrier that can sometimes prevent the outgrowth from a single cell into a clonal colony of cells, they've already overcome that barrier, so the efficiency and reliability of outgrowth is much higher. The bioprocess scientist is able to document and see as the cell, an individual cell, divides into two cells, then four cells, and then into a clonal colony prior to retrieving that colony directly off of the cell raft array. You are able to maintain an image-based record of that monoclonality, and you have evidence that the cells that you collect emanated from a single cell. Because you're able to screen at least 500 times more cells on a cell raft array compared to in a 96 well plate, this means that you're using a single consumable, the cell raft array, in the place of several 96 well plates. This also provides savings when it comes to media usage. If you think of 5 mils of media for a single cell raft array compared to 100 mils of media for just 5 96 well plates, you can isolate into fewer wells or plates on the back end. It really just depends on how many clones that you need. And this difference also comes to bear when you consider the cost differences between traditional cell line development and cell line development with the cell raft air. For a traditional cell line development workflow, if your aim is to generate 100 clones, the cost can be between $150 and $450 to be able to generate those clones. If you need more clones, say up to 500, this cost can go up sharply to close to $700 and up to $2,000. You're able to generate 100 clones for a cost closer to $75. If you're interested in scaling that up to 500 clones, the cost would be closer to $225. So you can see that even as you begin to scale up what your production needs are, the costs are compounding much more gradually when you're using a system that's intended to scale, like the cell raft array. One example that we're very proud of is from our customers at Kite Pharma. So Kite was interested in using CRISPR-edited Raji cells, so that's a suspension cell line. And what they did was they compared the cell raft array to several dispensers from companies like BD, Sony, and Thermo Fisher. And the outcome of this comparison was that they were able to get much more reliable clonal outgrowth when they used the cell raft technology compared to these sorting technologies.